Every species on Earth is constantly fighting the clock. Without a reliable mechanism to create new individuals, the final generation of any organism would simply die off, and that species would cease to exist entirely. That makes reproduction the single most critical biological process for survival. Looking at this split-screen flowchart, we start with asexual reproduction on the left. This is a highly efficient process involving just a single parent cell. Through a cellular division called mitosis, that cell divides to produce two offspring that are exact genetic clones of the original. On the right side of the chart, sexual reproduction takes a different path. It requires two parents. Instead of making exact copies, each parent produces specialized sex cells, called gametes, through a reduction division process known as meiosis. This equation illustrates why meiosis is necessary. Each gamete is haploid, carrying only half the normal number of chromosomes, represented by the letter N. When these two haploid gametes fuse, they restore the full diploid count, 2N, resulting in an offspring with a totally unique genetic mix from both parents. Asexual reproduction works perfectly if an organism just needs to multiply quickly, like a photocopier running off identical pages. But by continuously combining distinct genetic material, sexual reproduction generates the variation a species actually needs to adapt and survive when its environment changes. To see how plants execute this complex mixing of genetics, we have to look past their appearance. A flower is a specialized reproductive organ, precisely engineered to facilitate the exact transfer of gametes. This technical cross-section highlights the specific hardware involved. The large outer structures, the petals, serve as primary biological attractants. Their bright pillars and specific scents are built strictly to draw in biological agents like insects or birds. Moving inside, we locate the male reproductive hardware. The anther is the structure responsible for actively producing and housing the microscopic pollen grains, which contain the male gametes. Right in the center sits the female reproductive structure. The top part of this structure is the stigma, which functions as a sticky landing zone built specifically to capture incoming pollen. Every physical component we just mapped has a strictly defined job. A flower is a biological machine structured around one concrete objective, ensuring that male genetic material successfully reaches the female receptor. The process of getting that genetic material from the anther to the stigma is called pollination. It is crucial to note that pollination is purely a transportation phase. It is not fertilization. It is simply the physical logistics of moving pollen from point A to point B. This diagram maps the two possible transportation routes. The blue loop shows self-pollination, where pollen moves from an anther to a stigma on the exact same plant. The green arrow traces cross-pollination, where the pollen is transported across a physical gap to an entirely different plant. Plants physically adapt to their required mode of transport. On the left side of this split screen, we see a wind-pollinated flower. It lacks bright petals because it doesn't need to attract biological agents. Instead, it produces thousands of lightweight, micro-scale pollen grains designed to be easily carried by air currents. On the right side, the insect-pollinated flower uses a completely different strategy. It invests heavy energy into large, bright petals to attract a vector, and it produces much larger, sticky pollen grains, engineered to latch securely onto an insect's body. Whether relying on wind currents or a passing bee, a plant's physical anatomy perfectly matches the environmental vector it uses to transport its pollen. Once pollination is complete, the pollen grain sits on top of the stigma. But looking at this 1000x magnification on the right, we hit a physical block. The ovule, containing the female gamete, is located deep inside the plant, and the style separating them is completely empty. How does the genetic material bridge that void? The pollen grain actively solves this by growing a new structure. A microscopic pollen tube extends downward from the grain, carving a physical tunnel straight through the tissue of the style to reach the ovule. With the physical bridge complete, the male gamete drops down into the newly formed tube, traveling the entire length of the style while entirely shielded from the outside environment. The exact moment of fertilization happens right here at the base. As the male gamete exits the tube, it fuses with the female gamete. This microscopic collision forms a single diploid cell called a zygote. This precise moment of fusion is the finish line. The two separate sets of DNA are now locked together into a single organism, generating the genetic variation we identified at the very beginning. To correctly answer O-level exam questions on this topic, you need to sequence these specific mechanical phases without mixing them up. This chronological flowchart maps the exact pipeline. Step 1 requires identifying the anatomical structure. You must confidently distinguish which parts produce pollen and which parts receive it. Step 2 is pollination. Keep this definition strict. It is only the physical transfer of the pollen from the anther to the stigma, utilizing either wind or insect vectors. Step 3 is fertilization. This phase only begins once the pollen tube starts growing, and it officially concludes with the microscopic fusion of the male and female gametes to create the zygote. Pay close attention to these red warnings. The most common exam errors happen when students confuse pollination with fertilization, or when they incorrectly draw cross-pollination happening between two flowers on the exact same plant. Remember, the boundary for self-pollination is defined by the plant, not the individual flower. By keeping this biological pipeline strictly organized, from mapping macroscopic petals to tracking microscopic gametes, you have the exact sequence required to secure full marks and understand how plants maintain planetary biodiversity.